Hello, tonight we're going to talk about phylogenetic trees. This is going to be a real short video, so hang on, here we go. A phylogenetic tree is a representation of which organisms are related to which. So this is an example of a phylogenetic tree from a common ancestor. We've got different types of bacteria, fungus, the trees, the plants, and then the animals. Okay, so that's an example of a phylogenetic tree. The reason we use phylogenetic trees is to help understand how organisms are related. There's lots of different things that we use as evidence to build a phylogenetic tree. In the past, scientists used morphology, which would be physical and characteristics, anatomy, things like that. They'd use some behaviors and some biochemical attributes. Now we use much more than those. We use the DNA, the genome. How exactly are they related? Because the DNA is the evidence that demonstrates how closely organisms are related. Okay? So, in the past, we used something kind of like this, where we looked at what kingdom, phylum, class, order, family, genus, species organism belonged to. Okay? So, let's look at one specifically. Let's look at us. Okay, we are alive. We're part of the living part of the earth. We are in the domain Euchera, kingdom Animalia. Phylum Chordata, because you have a spinal cord, and actually a notochord. You are in the class Mammalia, the order Primates, family Hominid, genus Homo, species Sapien. So you are Homo sapien. Okay, that's our classification system, but that doesn't demonstrate how we're related to everything else. What other things are related to? So instead, we would look at something like a phylogenetic tree. Now, this is actually a genetic tree of the order Carnivora, which are carnivores. But you can see, going back very far, an Arctic fox and a kit fox are closely related, but the Arctic fox is fairly distantly related to something like a short-eared dog or a crab-eating fox. The reason I put this one on here, dogs are closely related to gray wolves, but they're actually fairly far distant related to other types of wolves. Bush dogs, maned wolf, and here's a little giant panda. Why not? Okay, and it even demonstrates on here, this separation occurred 6 to 7.4 million years ago. This separation occurred 3 to 4 million years ago. Okay, let's take a look at a little video, and then I'll tell you what your homework is.
Hi, it's Mr. Anderson, and welcome to Biology Essentials video number 6. This is on phylogenetics, and phylogenetics is essentially the evolutionary history or the evolutionary family tree of organisms. If you look on this page, we've got a number of pictures. We've got a number of pictures of cetaceans, so a bunch of whales. So the goal of phylogenetics is to create a phylogenetic tree. In other words, a tree that shows who's related to who. In other words, is the humpback whale most related to the gray whale or to the minke whale or to the fin whale? And so phylogenetics is actually a really fascinating area right now because with all the DNA evidence that we have, we're able to put together a wonderful picture. And our goal is that the phylogeny will match taxonomy. In other words, we can give names to organisms based on who they're related to. But the more you learn about this, the more you realize that all life is very, very similar. We have similarities between all of it. So let's get going on phylogenetics. And so basically, speciation is when one organism or one population or one group eventually diverges and so they can't interbreed anymore. It's the simplest way to think about speciation. And so phylogenetics and phylogenetic trees require speciation to have occurred. There are a number of different phylogenetics trees. The one that we'll talk a lot about are called cladograms, and they use what are called clades. and so it's just a specific type. But a phylogenetic tree shows the evolutionary history of an organism. Now, how do we figure that out? Well, we could use all the tools at our disposal. I'm going to talk about two specifically today. One is morphological. Morphological is the structure that you have, and the other is molecular. And so morphologically, I'm going to talk about hearts and how hearts have changed over time as they've required more and more things. And then the last is molecular. In other words, how do we use DNA to figure out who's related to whom? So those are phylogenetic trees. This is the evidence that we use to figure it out. And then at the end, I'm going to talk more about cladograms and how you create a cladogram. And so let's start with the biggest phylogenetic tree of all, phylogenetic tree of life. So if this is life down here, so if life began, 3.6 billion years ago, it's diverged into all these different lineages. And so this right here would be a phylogenetic tree of life. Now the one thing I want to point out, and Darwin was the first person to do this, is that whenever you have a tree, that suggests that there's common descent. And what does that mean? Well, bacteria, archaea, and eukarya, since they're all on the same phylogenetic tree, it means that they all came from that common ancestor. And so every time we have a branch point on here, so what does this suggest? That branch point right there suggests where that tree diverged into the eukarya and then the archaea that we have. And so the idea of descent is a long one, but the more evidence that we gather, the more we realize that Darwin was right on. Now we have to figure out who's related to who. And so let's start at evidence that we have. So the evidence that we can use to make a phylogenetic tree. Let's start with the first one is morphological. Morphological are the structures that you have. And so this is a phylogenetic tree of vertebrates. And so we've got early vertebrates, we've got time periods over on the side, but we're most interested in the mammals. We've got birds, reptiles, amphibians, fishes. And so how did scientists figure out who's related to who? Well, we can choose one characteristic and then we can trace that through time. We can look at one thing and see how it's changed over time. So a perfect example would be the heart. The heart began in fishes as a two-chambered heart. A two-chambered heart really just has one valve. In other words, the blood is going to flow in this direction, and then there's a valve that opens in this direction. So once the blood moves through it, it can't come back in. And so it's just a muscle that has a valve on the inside of it. And so what's the function of the heart? Well, in a closed circulatory system, in other words, insects don't use a closed circulatory system. Their blood just goes everywhere in through the tissues. But in a closed circulatory system in a fish, the blood, which in blue in this case just means that it's deoxygenated, is going to go through the gills, and then it's going to be oxygenated, so it's red, and then it's going to go through the body and the tissues in the body, and then it's going to drop off that oxygen, and then it comes back to the heart again. And so a two-chambered heart, a better way to think about what a two-chambered heart is it's simply a single loop. So we just have this one loop through the gills and back to the body. And for fishes, that works great. The problem is that as we move on to land, there are quite a few more constraints as you move, especially as you move towards being like a warm-blooded organism. And so the constraints get heavier and heavier. And so it's okay to have a two-chambered heart. It works great if you're a fish. But as we move on to land, then it has to modify itself. And so again, we just have this one loop. The major problem is that once it goes through the gills, you lose a lot of the pressure. And so you lose the pressure, and so it's hard to move that through the rest of your body. It works great if you're floating in water, but as you move on to land, we don't have that pressure. Okay, so let's go to a three-chambered heart. Now, a three-chambered heart arrives in the amphibians, and so things like a frog have a three-chambered heart. So they've got three delineations. We've still got that loop that goes through the lungs, and mostly in amphibians it actually goes through the skin where they pick up oxygen. But you see that we now have a problem here. We're not losing that pressure. In other words, we're able to pump the blood to the skin in the lung, or excuse me, we're able to pump it to the skin in the lungs, and then we have a separate loop that goes through the body. So we still don't have to deal with that pressure. But the problem comes in right here, and that is that we have a mixing of the oxygenated and deoxygenated blood, and so it's purple. Now, is that a problem? Well, it's a problem if you are anything above or spend more time on land than amphibians do. And so it works great for amphibians, but you have a mixing of oxygenated and deoxygenated blood. And so if you look at this loop, it's a double loop, but if you look at it and say, okay, now let's move up to the reptiles, and now we have to move more blood, and we don't want as much of this mixing here because we're going to lose a lot of that oxygen. Well, think about it as an engineer. How could you solve this? Well, a three-chambered heart works like this, and what it does is it has a septa that's built right down here in the middle of the heart, and that septa separates the deoxygenated from the oxygenated. It still has a little bit of mixing of the blood, but that works great because they're cold-blooded critters, and so as they move that blood around their body, they can actually keep themselves a little bit warmer. But that's a three-chambered heart, and as we move on to endothermy, as we move on to birds and mammals, that just doesn't cut it. And so we think that birds and mammals both evolved this independently, and you can see on here that birds branched off from reptiles and mammals branched off earlier from a common ancestor. And so we eventually have the arrival of the four-chambered heart. What's the four-chambered heart do? Well, you can see that septa that went right down the middle has completely closed off, so we don't have any mixing of the oxygenated and deoxygenated blood. And so birds and mammals have this morphological change, and they did it because they're endothermic. In other words, they require a constant body temperature. And so we can trace this morphological evidence through the organisms. We can say who's related to whom. In other words, if we have a three-chambered heart that's shared by everything above here, that means that on our phylogenetic tree we want to at least put those on the same branch. Next, I want to talk about molecular data. So molecular data is looking at the DNA, so looking at the genetic code. And so this is a study that was done in 2009, and what they were trying to figure out is where metazoans fit and who's related to whom. And every time I have a new biology book, I find that this is actually organized a little bit differently. But we have this group down here of the jellyfish and the sponges down here on the bottom, and then we have this group up here which contains things like us. And so scientists weren't sure if this branched off early or if these branched separately. And so what they did is they gathered a huge amount of DNA evidence. And so you can see here that this was a very large study done on a number of different families, a number of different groups of animals. And they looked at mitochondrial DNA, proteins, ribosomal RNA. They looked at a number of different things. And they figured out, this is very recent, that this branch and this branch are actually sister branches. The branch up here that makes us and the branch that makes the jellyfish have kind of separated a long time ago, and they've been evolving since then. And so this is a great way, this would be a phylogenetic tree that we can use molecular evidence to answer a problem. But you have to gather a huge amount of data before you can actually do that. And on here you can see that they actually have an outgroup, which is a group of fungi, which is not an animal, but it's a way that we can actually make comparisons to that molecularly, and then we can figure out the connections. Okay, last thing I said I'd talk about are cladograms. Cladogram uses what is called a clade, and a clade, if I remember right, comes from, I think Latin, it means a branch. And so a clade is simply a group that has an organism and all, and I would circle the word all, of its descendants. And so this right here is a clade because it has this organism and all the descendants that come from that. Or this, the orange, is going to be a clade because it has this organism and it has all of the descendants that come from that. But green right here, we would not call this a clade, and the reason why is that you would have this organism right here and all these descendants, but you're missing a number of them over here. And so that's not a true clade. And so cladograms are going to, it's the definitive answer of who's related to whom. But you use two things. You use molecular and DNA for sure evidence, but the other thing that you're going to use are something called synapomorphies. And a synapomorphy is going to be a characteristic that's shared by all of those in the clade. So a couple of good ones, as we look through the fossil evidence of dinosaurs, we branch all dinosaurs into two groups, the Ornithischia and the Saurischia, and these are all, Saurischia, if I remember right, means bird-hipped, and Ornithischia means lizard-hipped. And so it's a way to branch these groups, and so a synapomorphy would be this characteristic. In other words, Saurischia is going to be in this hip structure, is going to be shared by everything in this clade. And so the goal is to have a clade that has similar characteristics, and it also doesn't leave anything out. A real example that you're probably familiar with would be reptiles. Reptiles is a silly term, because reptiles used to be this blue area right here. It contained things like turtles, it came crocodiles, and birds we left out of that. And so if you look at this group is what's called paraphyletic, and so reptiles as a group was paraphyletic. It had all of these descendants, but it lacked the birds, and we now know that birds are a part of this group. And so the goal of a cladogram is to create what are called monophyletic groups. Monophyletic would be this, would be this yellow group right here, because it contains this and all of the descendants of that move from here. So polyphyletic means you have groups that come from different areas. So if we were to put mammals and birds together in one group, that'd be polyphyletic. And paraphyletic is when you have some organisms but not others. And so what's the goal? The goal of a cladogram is to figure out all of life. So we put all of life on branches, and we figure out who's related to whom. And then hopefully we can use a naming system and classify all of life. It seemed like a daunting task at one time, but molecular evidence is giving us an inroads to that, and it's a really hot topic as far as biology goes today. So that's phylogenetics, and I hope that's helpful.
I hope that video was helpful to you. Okay, so what I want you to do now is go to your bioportal and complete all of the activities for Chapter 16. That's your homework. Okay? Have a nice night. Bye.